Thank you, Dawo, for highly personalized introduction. I'm flattered. And Khawala. And thank you to you and Professor Shattuck for organizing these seminar series. It's a huge honor to present as a part of these series. And I imagine, a huge amount of work that goes into running these every single day. But also thank you for providing us a chance to escape to our data sets and our papers away from currently quite worrying reality that surrounds us globally. So thank you. I would like to use this opportunity today to talk about, even ask a question, is there a causal link between intracellular sodium elevation and cardiac metabolic remodeling? And even though currently I am at Queen Mary, this is actually work I have done at King's College London, here at St Thomas's Hospital, the Rain Institute, and this quiet picture could as well have been taken today, given the current lockdown state in London. So my background is in cardiac metabolism, and cardiac metabolism in the heart is fascinating because the heart is a #1 ATP consumer. It is a very mitochondrial rich organ because it has a huge demand for ATP. It goes through 6 kilograms of ATP per day, all to support varying cardiac workload. And as such, the cardiac metabolism is a complex machinery in terms of regulation, all to support this enormous workload. We can really divide both regulation of cardiac metabolism into substrate uptake, cytosolic processing of substrates, oxidative phosphorylation, where most of ATP is synthesized, and energy communication pathway mediated by creatine kinase that mediate the energy reserve in the heart. So the healthy adult heart relies heavily on fatty acids as a source of ATP. They are very ATP rich, and secondary to fatty acids is glucose. And it's this heavy reliance on fatty acids over glucose that we define as healthy adult cardiac metabolic phenotype. However, over the past century, we have known that it is the changes that we have changes in cardiac metabolism in hypertrophy and failure. And with the advent of improved technologies, including magnetic resonance spectroscopy, we're now able to really stratify these metabolic alterations that occur in heart disease and to split them between substrate utilization, intermediary metabolism change, and also changes in energetics that lead to energy starvation and heart failure. So there's a switch in this substrate preference in adult heart from fatty acids to glucose, and this happens in disease process. And we define this switch as switch from from adult to fetal metabolic phenotype. And this is initially adaptive process. However, long-term wise, glucose is insufficient to provide sufficient ATP from varying workload of adult heart. And as shown in seminal work by Stefan Neubauer and colleagues back in the 90s, we have actual derangement or complete disappearance, in fact, in some cases, of the cardiac energy reserve, phosphocreatine to ATP, shown beautifully here in a healthy volunteer. This is in vivo phosphorus human spectrum, to complete disappearance here seen in a dilated cardiomyopathy patient. In fact, this patient was dead a few days within this spectrum being acquired. So it goes from change in substrate preference to complete derangement of the system, where we get energetic failure. So this sort of energetic or energy deficit theory that the failing heart is engine out of fuel. So in addition to all these metabolic changes that happen in heart disease, cardiac hypertrophy and failure are also characterized by sodium overload and impairment in EC coupling process. So sodium has been measured in various species, from humans, healthy human heart, diseased human heart, large animal models such as sheep and pig, down to mice, which we use extensively in the labs. And even though sodium values may vary depending on technique people use to quantify sodium, there's really one common agreement by now and conclusion that in both human and animal models of hypertrophy and failure, we have chronic sodium elevation. But what causes it? So the key player in sodium homeostasis and cardiomyocyte is the sodium-potassium ATPase. Why? Because there are many ways that sodium can enter the cardiomyocyte, but there's one principal mechanism for sodium efflux, which is via sodium-potassium ATPase. That's why sodium cardiac, sodium potassium, that's why sodium pump plays such a crucial role in transmembrane transport, ion homeostasis, electrical excitability, as well as the control of cell volume. The sodium pump itself is regulated by a protein, phospholemon, which is a member of fixed family of proteins. So it's a phosphorylation or dephosphorylation of phospholemon that regulates the sodium pump activity. And this phosphorylation is mediated by PK and PKC signaling pathways. So many ways for sodium to get in, but really one principal way for sodium to leave the cell. However, it's really not a black and white story, because there's still ongoing debate. What is the key mechanism behind this sodium overload, which we see in models of heart failure and hypertrophy? Is it increase in influx pathways of sodium? Or is it a sodium pump inhibition, or just an inability to sufficiently remove sodium from the cell? And work by my colleague Andrzej Bogusławski in Professor Shattuck's lab has in fact shown that in a mouse, which I will also use in a study I'm about to present, it's a mouse that has chronically unphosphorylatable phospholemon, therefore sodium pump is chronically inhibited. He has found that chronic sodium pump inhibition results in increased susceptibility to hypertrophy-induced dysfunction. So it could be that it's inhibition of sodium pump that could be a key player in the sodium overload that we get in pathological states. So we know that we have change in metabolism, and we have sodium overload and hypertrophy and failure. But there's always this question, what comes first? In fact, what's the relationship between these two mechanisms? Because sodium pump is very ATP hungry. It actually consumes quite a decent chunk of ATP synthesized in cardiomyocytes. And if you look at the metabolism literature, there is this prevailing almost assumption that it's purely because of this metabolic remodeling and energetic deficit that pump just doesn't get enough ATP, and therefore the pump slows down and sodium accumulates. But this isn't necessarily true, because if you look at the biochemical literature, never mind just pure cardiology literature, we can actually see that the delta G and concentration of ATP, even when they're low, do not necessarily inhibit sodium pump. And in a beautiful study by George Rada in the mid-90s, it's shown that in low flow ischemia, when ATP is low, it's still not low enough to abolish completely the activity of sodium pump. One sparked our interest is also work done in the early 90s by Cox and Matleb in isolated cardiomyocytes and organelles, where they have shown that increase in cytosolic sodium activates mitochondrial sodium-calcium exchange, and therefore reduces mitochondrial calcium, which could potentially affect activity of all mitochondrial calcium-sensitive dehydrogenases, which are key players in TCA cycle and oxidative phosphorylation. This includes pyruvate dehydrogenase, isoleucine dehydrogenase, and alpha-ketoglutarate dehydrogenase. So this potentially, they have postulated that this high sodium, by activating mitochondrial sodium-calcium exchange, can, in essence, cause energetic deficit by slowing down these calcium-sensitive dehydrogenases. But again, this was the work done in cells and organelles, and this was assumption that's never been actually shown. So again, the extent to which metabolic changes reflect chronic cellular remodeling, or whether they arise as a consequence of sodium elevation, have not been determined. So this led us to ask a question. Is there a causal link between acute and chronic sodium overload and metabolic remodeling in the heart? And we decided to put this traditional thinking on its head. What if it's actually metabolic dysfunction, i.e. the mismatch in ATP supply and demand, that is secondary to sodium pump dysregulation and intracellular sodium accumulation? And also another question is, does pathological hypertrophy share a common sodium-mediated metabolic fingerprint with acute and chronic sodium load? Excuse me. So we have decided to use integrated experimental approach in beating Langendorf perfused hearts. So this is a typical Langendorf benchtop apparatus, which people might have seen in labs across the world. And we have adapted that to a completely MR-compatible umbilical Langendorf perfusion system, as shown here. So that enabled us to play. No. Yeah, so we have perfused mouse heart here, an NMR tube, which allows us to perfuse heart at physiological temperature with crystalloid KH, and we can drop it into the core of the magnet, 9.4 Tesla magnet, and we can real-time monitor cardiac function, perfuse lung of cardiac function, as well as acquire a whole host of biochemical measurements in terms of different types of NMR spectroscopy. And we have also adapted this umbilical system to be able to rapidly exchange our buffer solutions without any sort of dead space or dead volume, so we can use a whole host of pharmacological modulations in our study of sodium, impact of sodium on metabolism. However, system was there, we could perfuse hearts, but given that we wanted to examine this relationship between intracellular sodium and metabolism, we actually had one problem. Intracellular sodium beating heart has been very successfully measured over the years using shift reagents. And these work by purely shifting this huge extracellular sodium signal, which is the order of magnitude of about 140 millimolar, to expose this tiny intracellular sodium peak, which is about 8 to 10 millimolar. And after you start infusing these shift reagents, which are chelators, into the heart, after about 10 minutes, you shift completely the extracellular sodium and sodium in the buffer surrounding the heart, and you get this tiny intracellular peak, which, like I said, works really well. The problem is these chelators also chelate all the calcium in the buffer, so you lose all your function. So you may get a very nice sodium measurement in your perfusion, but you also end up with a piece of dead meat because the heart is not beating anymore. So that was no good for studying the relationship between sodium and metabolism. And we have been very fortunate that in imaging department at King's, we had a talented chemist, Dr. Thomas Eakin, who helped us to set up and validate the new approach to measuring intracellular sodium in perfused heart, multiple quantum filtered sodium spectroscopy. So we have first embarked on setting up this multiple quantum filtered or triple quantum versus double quantum filtered sodium spectroscopy, which relies on the behavior of sodium in a magnetic field to, in semi-quantitative manner, measure intracellular sodium. And we went through a series of studies first validating correlation between TQF-DQF ratio and intracellular sodium load measured by more traditional techniques. So only when we're happy that we can measure sodium in perfused beating heart, we were then ready to move on to studying the metabolism, relationship between sodium metabolism in our question. So like I said, we use integrated experimental approach. So in addition to sodium spectroscopy, our perfused hearts, we also did phosphorus spectroscopy to assess energetics. We did high resolution proton metabolomic profiling using proton spectroscopy. Hearts were freeze clamped at the end for this thorough metabolomic analysis. And also we set up a validated carbon-13 steady state spectroscopy to measure substrate preference in a perfused heart and metabolic flux. And also in collaboration with colleagues at Birmingham and Merck Sharp and Dohme, we also did some mass spec of our frozen hearts at the end of the protocols. So the first question was, how does the chronic sodium elevation impact cardiac metabolism? In order to study this question, we used two models. First model was the animal model, transgenic model of phospholemon 3SA mouse, which is the mouse I briefly mentioned in my introduction. It's a mouse with unphosphorylatable phospholemon because of this serine to alanine mutation, which renders the pump chronically inhibited. And second model was more clinically relevant model. It's a model of hypertrophy that was well established in the lab, which is TAC banded surgery, 5 weeks, in fact, post-TAC. So we had two models of chronic sodium overload. So both models, myocardial sodium was elevated to similar extent in both perfused hearts, cardiomyocytes, and you can see here both from TQF-DQF spectroscopy as well as a total sodium measurement. So we had two models of chronic sodium elevation. This was the actual experimental protocol that was used. So after the hearts were calibrated with crystalloid hyperglycemic KH, we were acquiring a whole series of baseline phosphorus spectra and sodium spectra. And then we would continue the 30-minute perfusion, again, continuously acquiring sodium spectra because acquisition time is very short. It took about a minute and a half or so, and phosphorus spectra to build a full picture of sodium and energetics in these hearts. At the end, the hearts were rapidly freeze clamped for proton spectroscopy and mass spec. And then we have repeated all of this with full metabolic KH, where we supplemented are perfused hearts with fatty acids, glucose, lactate pyruvate, glutamate, and insulin to sort of mimic physiological concentration of substrates in a mouse blood. At the end, we rapidly freeze-clamped hearts for carbon-13 and NMR spectroscopy analysis. So what we found is that with chronic sodium elevation, we didn't have energetic impairment. So there was no reduction in PCR to ATP ratio or evidence of ischemia or change in pH in both of our models. So both phospholem and 3SA mouse, as well as the model of hypertrophy. So no energetic impairment with chronic sodium overload. However, we found something else which we thought was fascinating, is that hearts with chronic sodium can maintain their ATP, PCR to ATP reserve, but we have a switch in substrate preference from fatty acid to glucose utilization. And this was the same pattern of change in both phospholem and 3SA sodium overload and pathological sodium overload. We had reduction in fatty acid utilization and increase in glucose oxidation in both our groups. Going back to our initial background sort of theory of mitochondrial sodium-calcium exchange, we then used mitochondrial sodium-calcium exchange inhibitor, CGP137157, to acutely inhibit the mitochondrial sodium-calcium exchange in our protocols. And what we found is that even this acute inhibition of mitochondrial sodium-calcium exchange reversed metabolic remodeling. So this switch in substrate preference from fatty acids to glucose observed in both models of chronic sodium overload was readily reversed with inhibition of mitochondrial sodium-calcium exchange. In terms of the metabolomic profile, the chronic sodium elevation caused a global depletion of energy-providing substrates in 3SA, phospholem and 3SA mouse, including the intermediates of TCA cycle, amino acids, and lipid metabolism constituents. And again, we have managed to completely revert, to rescue this phenotype with CGP treatment. Our hypertrophied hearts, we were talking about at the compensated stage of hypertrophy because there was no energetic deficit. So we didn't have as much of severe metabolite depletion profile. However, again, any change we did observe was very much rescued with CGP in a similar fashion that we saw in 3SA mouse. So that was the impact of chronic sodium elevation. We then examined the impact of acute sodium elevation on cellular metabolism. So for this, we have used pharmacological sodium pump inhibition using warbane, which is a classic sodium pump inhibitor. But because it causes positive inotropy, we have also titrated blebistatin into our perfusion preparation to uncouple any sort of positive inotropy, which allowed us to measure just the effect of elevated sodium metabolism without any increased energy demand. So this was a typical perfusion protocol of use, equilibration. Again, we had the baseline acquisition of sodium and phosphorus spectra, and then we elevated sodium and acquired phosphorus and sodium spectra, froze the hearts at the end for metabolomics, and repeated this type of study again with full metabolic buffer for C13 substrate utilization assessment. What we found is that when we infused with this acute pharmacological treatment of perfused hearts with warbane, increased sodium readily. So you can see here that warbane and warbane blebistatin perfused hearts have dramatically elevated sodium versus baseline. And most importantly, if you look at the LVDP plot, is that warbane and blebistatin perfused hearts have exact same LVDP as control hearts. So they have a high sodium with normal LVDP, so no increased energy demand. What we found with acute sodium elevation is similar changes that we saw in the chronic elevation. Energetics was again maintained, so there was no reduction in PCR to ATP ratio. So these hearts were not energetically compromised. And there was again reduction in beta oxidation of fatty acid utilization and increase in reliance of other substrates, including carbohydrates. And with acute sodium elevation, because the sodium elevation was more severe than the chronic model, we had more severe depletion of metabolic intermediates, including TCA cycle intermediates, acetate and aspartate. And we have repeated our acute sodium elevation experiments again with the CGP inhibitor, and Same as in chronic group, we found that the changes, the severe depletion of metabolites we observed with acute sodium elevation, this was completely ameliorated with the mitochondrial sodium calcium exchange treatment. We have gone one step further in terms of trying to piece together a picture of the common metabolic changes, or generally trying to build an idea of metabolic hallmark of sodium elevation. So we have approached Dr. Anja Karlstadt and Dr. Heinrich Tegmer, University of Texas. Anja is an expert in silico modeling, and she has this fantastic CardioNet model, computational model of human metabolism. So her model can look at over 1,700 metabolic reactions in the cell across different cellular compartments. And she has used our experimental data from various metabolism experiments we have done to try to tease out the common changes in metabolism between our chronic and acute sodium elevation groups. And Anja's work has beautifully shown that we have distinct change in metabolism between our control and sodium elevation groups. This is a principal component analysis plot of Anja's CardioNet metabolic in silico model data. And she has also shown that in agreement with experimental work, that we have this common fingerprint of metabolic derangement with sodium elevation, reduction in fatty acids, and enhancement of carbohydrate metabolism as adaptation to high sodium to maintain ATP supply. This is a simplified heat map plot of Z scores that Anja did, because the full one is over 700 reactions in size. But really take-home message is that with acute sodium elevation, we have reduction in fatty acid utilization, and as a result, we have enhanced metabolism of pyramide lactates, amino acids. And with chronic sodium elevation, we have reduction in fatty acid oxidation, same as in acute, and enhanced glucose and ketone utilization. So we have remodeling or jiggling of metabolism to maintain ATP supply in a similar fashion in both acute and chronic sodium groups. So in conclusion, we have seen that sodium overload, whether it is acute, whether it's chronic due to transgenesis or hypertrophy, results in common metabolic perturbations. And most interestingly, they seem to precede any energetic impairment. So we have switch in substrate preference from oxidation of palmitate to oxidation of glucose. We also have severely altered metabolomic profile in terms of TCA cycle intermediates, glycolytic amino acid intermediates, however, without energetic impairment. And which I thought was most exciting is that even acute inhibition of sodium calcium mitochondrial exchange by antagonists seems to reverse these changes in all the groups we examined, regardless of etiology and duration. And our in silico modeling has indicated, again, common alterations in terms of change in fatty acid metabolism, carbohydrate, and amino acid metabolism. So we believe that these changes constitute a metabolic hallmark in response to sodium elevation, and they are independent of etiology or duration. And collectively, we believe that these observations suggest that perhaps early prevention of myocardial sodium elevation could either prevent the origin or alter the course of metabolic derangement in pathological hypertrophy. And on this note, I would like to thank my colleagues. First of all, Professor Michael Shattuck, PI in this project, and person responsible for introducing me to the sodium field from a pure metabolism sort of world. It's been an amazing experience for the past 7 years. Tom, without your help with the sodium spectroscopy, none of this work would really be possible. Dr. Tegmer and Anja for all their metabolic modeling work. Our colleagues in Birmingham, Daniel Tennant, Alpesh Tucker, Davor Pavlovic, Will Fuller, University of Glasgow, all my colleagues from Shattuck Group and British Heart Foundation for funding our work. And I would like to leave you with the view outside St. Thomas's Hospital, because no matter how good or bad metabolism experiments are in a day, walking out to this view every day has been a huge privilege over the years. So thank you very much, Davor. I'm happy to answer any questions.
Well, thank you very much, Dunja. Do you want to maybe stop sharing the screen and then...
Yes, I'll do that now.
Does
that work?
Okay, yeah, perfect. So effectively now we are back to Q&A. So if you would like to maybe just look through the Q&A, if you click on the Q&A box.
I did. I'm looking through Exchange here.
Mike is meddling. Mike, stop meddling.
Thank you, Mike. It's a call a friend situation. Right, so I can answer these questions, save Dunja the trouble. Is this for the first two questions? Are there other functions of phospholemon?
First question. So maybe you want to just read it out.
Okay, so Brian Martin says, are there other functions of phospholemon other than sodium-potassium regulation? Sodium-potassium expression is reduced. Jamin Chen says sodium-potassium expression is reduced in hypertrophic heart. How about expression of phospholemon? Does it increase due to a compensatory mechanism or decrease? Which is the expression of NCX in mice with chronic inhibition of sodium-potassium HPAs. Does intracellular and sarcoplasmic calcium concentration increase? So Mike has answered these questions. There are reports that phospholemon regulates sodium-calcium exchange. However, this has not been reproduced by others, nor is NCX believed to be phosphoregulatable. So my answer is no, not to my knowledge. Phospholemon, at least in muscle, seems simply a regulator of NKA. It links signaling to sodium transport.
Sorry to interrupt you. Have you just skipped? Because I think Mike's only answered the first one.
Okay.
From what I can see.
Okay, so I can see question from Gary Lopeshik. So after Mike's reply about NCX slightly increased in 3SA mice. Yes, Alicia, actually my colleagues, Andrzej Bogusławski's 2014 paper in Cardiovascular Research, where he's the first published the phenotype of phospholemon 3SA mouse, I believe he has actually looked at the expression of a whole host of proteins, including NCX. So full details of the changes in protein expression, including increase in NCX, is actually described in that 2014 paper, which I referred to at the introduction of my talk. So Gary Lopeshuk, acute sodium overload appeared not to increase glucose oxidation, but rather the contribution of unlabeled substrates. What do you think these unlabeled substrates are? Thank you, that's a very valid question. The unlabeled substrates that we used were pyruvate, lactate, so other carbohydrates, pyruvate, lactate, and glutamate. I think lactate is a good candidate because I also saw increase in lactate levels in the hearts of acutely elevated sodium. And I think Professor Lopeshuk might have actually had this discussion with you around the meetings about this, the lactate shuttle mechanism and uncoupling of glycolysis to start using lactate rather than just effluxing lactate from the cell. So I think lactate is a very good candidate for the use in acute sodium group. And this is also something I've seen in my rat work in a different model of hypertrophy, where I saw lactate being a #1 carbohydrate, actually preferred over glucose in perfused rodent heart. So I haven't labeled lactate in these experiments. In my previous work, I was doing dual perfusions where I did palmitate label, and one set of perfusions will label lactate, and separate set of perfusions will label palmitate and labeled lactate, and I was able to tease out this specific contribution of individual carbohydrates. I haven't done that this time, but my bet would be lactate, which based on my metabolomic levels. I hope that answers your question. Thank you, very nice talk. Can you detail again why fatty acid oxidation rather than oxidative glycolysis is affected upon this elevation of sodium? Matthias, you ask a very valid question. My study has identified substrate switch, but my study has not. I haven't pinpointed mechanism in terms of the crosstalk between substrate utilization, where exactly things change with high sodium. This is the first study we have done, and the future work is going to look to tease out this mechanism better. Sodium pump has a preference for glucose or glycolysis. So I think historically looking at also metabolism papers, there always seems to be enhanced glycolysis associated with changes in sodium pump activity. This could be one of the reasons, and also because it can be used both anaerobically, aerobically, and in cytosolic. So it's close. Spatially close to the ATP provision from glycolysis is closer to location of the sodium pump in the cell. Only used inhibitor for sodium calcium. So Hector Chapoy, sorry, only used inhibitor for sodium calcium mitochondrial. But what's up with MCU? This calcium could activate the TC enzyme. Well, I haven't looked at the MCU activity. I have looked at the some of the protocols for a perfusion protocols, MCU inhibition in type of experiment I have done. And the evidence I've seen from literature, from papers, is the type of inhibitors that we use when not going to necessarily dissect the mechanism that I was after. So we're very much looking into this. Like I said, this is the first, hopefully, of many studies trying to dissect or tease out the mechanism of the sodium metabolism crosstalk. Kenneth Fish, if cardiomyocytes can be isolated from the perfusion experiments, especially those that you have NMR data for, it would be very informative to look at changes in transcriptome. Resulting data could be correlated with in silico data to validate the modeling results. Sequencing may be more informative than proteomes, since the rates of changes of transcription usually occur on the same scale, I presume, some of these experiments. That's a valid suggestion, maybe for chronic experiments. I'm not sure if my short acute sodium elevation protocol is long enough to induce this type of changes, but maybe my chronic groups, perhaps, yeah, you're right, worth definitely thinking about. Oh, this is Mike answering. Thank you, Mike.
Mike Madeline. Dunja, may I just interrupt here and just kind of respond to that, basically, because I think what Mike is trying to say is just that underphosphorylation of phospholemon is the main observation rather than the expression of the pump, which therefore reduces the pump activity. That's all. So.
Thank you, Davo. Eduardo Ramen says Philadelphia. Great. Ivana, thank you. What is the possible therapeutic potential? Well, like I said in conclusion, we believe that sodium pump, modification of sodium pump, or activation of sodium pump rather than sodium pump inhibition could have therapeutic potentials with a compound similar to the one that we used in our study. However, there's a cautionary tale, obviously, of targeting a sodium activity, because we know cardiac glycosides have been now used for 200 years, but they've been inhibiting the pumps, therefore making matters worse because they are perceived to be energetically increasing energetic cost of already struggling the heart. So perhaps I think ideally we would have sodium pump activator compound that would activate the pump to release some of this sodium load that cells tend to accumulate with the process of remodeling. So that would be a really ideal drug would be sodium pump activator rather than any further sodium pump inhibitors. What is the phosphorylation status of phospholemon during these experiments? I actually haven't done Western blot for every, I mean, this mouse, first of all, the 3SA mouse has unphosphorylatable phospholemon, but in terms of my acute group, to be honest, I haven't looked at the Western blots of phospholemon phosphorylation. Do you expect similar metabolic changes secondary? So Kishore, sorry, Pastor Marty, do you expect similar metabolic changes secondary to increase in blood volume and sodium concentrations? A situation similar to heart failure? It's an interesting question. And the sodium overload, this is close to my heart because actually my PhD work was done on uremic cardiomyopathy, the model of renal failure, where I had consistent sodium volume overload as well as pressure overload in rats. And this initially, it's actually this PhD work that sparked my interest in sodium pump. And that's how my conversation with Davor and Professor Shattuck started. I haven't looked at this. I would love to repeat these experiments, which I've just shown here, in the model of chronic sodium overload and volume overload, such as uremic cardiomyopathy. The work in uremic cardiomyopathy has shown changes in metabolism similar to what we observed in a study here with the phospholemon 3SA mouse and acute sodium overload. So I would love to repeat this work using the tools that I have at my disposal now with all these techniques I've described to look at the uremic cardiomyopathy where I have high sodium and volume overload. So I haven't done the work yet. I hope hopefully something that we can do in the future. David Eisner, I agree that stopping the rise of intracellular sodium will stop the metabolic changes, but won't be negatively inotropic. Perhaps not a good idea. Well, Professor Eisner, I'll take your advice. Eduardo Rame, hello, great talk, Dr. Aksentijevic, thank you. The observation in chronic sodium overload are of interest as you also observed an increase in ketone substrate utilization. We described the metabolic adaptation to ketolysis in chronic advanced human heart failure, and I'm interested in your or Dr. Tegmeyer's explanation for the observation that this observed in chronic but not acute. Thank you. It's an interesting question. I mean, when I think of the ketone body utilization, I have to think about the global metabolic phenotype because even though ketone bodies are going to be closely correlated to fatty acid metabolism, I believe that their availability globally is going to play an important role, as well as in cardiomyocytes. Saying that, I haven't observed systemic metabolic change in our chronic sodium mouse model, because I have done the whole serum profiling and I haven't seen any changes in circulating ketones or other metabolites. So the only thing I can think of is that the ketones are being used because they are oxygen savvy substrates to be used at a time where heart is clearly remodeling, rejiggling metabolism to keep ATP synthesis, because even though I kept re-emphasizing during my talk that we didn't see the evidence of energetic deficit, the chronic sodium elevation, still these hearts are highly metabolically remodeled. So it could be the case that they're using anything, any substrate, anaporotic substrates, other substrates that can provide ATP to keep the flux maintained. So I'm sorry if I don't have more detailed explanation of ketolysis. I definitely didn't see change in circulating ketones in this chronic sodium overload model. And in terms of the utilization at the cardiomyocyte level, I can only comment that they basically stepped in to provide ATP synthesis at a time where metabolism was being severely remodeled. And Rosanna Bassani, is there any change in ATP consumption in your models? What about cytosolic pH? No. I had 30 minutes today, therefore I had to prioritize which data I have shown, but I have checked, given that we've done sodium spectroscopy in all our metabolism, in all our sodium experiments, I haven't seen drop in pH and any sign of ischemia in hearts. And given that we have maintained cardiac work because the function was normal and that PCR to ATP ratio is maintained, I think ATP provision is normal, ATP consumption. Well, I specifically introduced blebbistatin in my acute sodium group not to increase ATP consumption by the hearts, to keep it steady. So I'm not sure if that answers your question. Fatima, a great seminar, Dr. Dunja, thank you. Do you look at expression activity of the sodium protein isoform? No, I didn't. But Andrew's paper might have looked at the NH trisoform 1, but no, I haven't in this study.
I'm not aware of it, actually.
No?
Don't think so.
You mentioned the lactate used rapidly in heart failure models. Does that mean that the glycolysis is a part of glucose metabolism rather than mitochondrial? So does that mean that glycolysis is a part of glucose metabolism? Well, it is, rather than mitochondrial glucose oxidation in failing heart. Do you mean cytosolic glycolysis versus the full glucose oxidation, entry of pyruvate and TCA cycle oxidation? I'm not quite sure what this what the question is. In terms of lactate production, I did not see switched anaerobic glycolysis in my chronic sodium models. In fact, not in my acute sodium model, because we don't seem to have a switch from oxidative phosphorylation to anaerobic glycolysis. That I do know because I have looked at the effluent lactate from my perfusions. So I'm not sure so it's not anaerobic cytosolic glycolysis. It seems like glucose is going fully through its oxidative metabolism of glucose. Thank you, very interesting talk. Another phenotype of seeing someone failing heart is an increase in stiffness. Do you know if phospholaminin? So
this is a question from Joseph Huntington. Oh, Dunja, you've just frozen.
Sorry, no?
You've frozen for a second, so maybe you just want to go through that again.
Oh, sorry. Yes, Joseph Huntington is asking. Very interesting talk. Thank you, Joseph. Another phenotype of seen in some failing heart is an increase in stiffness. Do you know if phospholaminin or sodium potassium ATPase is regulated downstream of mechanical stimuli, and if this mechanical stimuli will affect the metabolism? If the sodium potassium ATPase and phospholaminin are regulated downstream of mechanical stimuli, I don't know the answer to that question, I'm sorry. But obviously any change in mechanical stimuli or increase in demand is going to affect the metabolism because it's going to increase the ATP demand. So, I mean, I haven't done heart space at the heart superphysiological rates or anything like that to be able to answer this question. So sorry, I can't help with that one. Daniel Johnson, very interesting talk. Thank you, Daniel. Do you know if there are differential pathways in atrial versus ventricular tissue in the link of metabolism and increased in sodium? I haven't looked at the difference between atrial and ventricular tissue. I have done the whole global cardiac perfusion, so I haven't dissected atrial versus ventricular tissue, I'm afraid, at this stage. Kenneth Fish, love the talk, congratulations, thank you very much. Brian Martin, are there changes in any isoforms of sodium, potassium, phospholemon that could change dynamics of the channel? This is the bit where I need the mic to Mike's cheat sheet to answer this question. I'm not sure what the answer is, I'm afraid. Sorry, Brian. Karzubia, very nice novel data. Thank you. Thank you very much. Using the CGP compound, were there improvement in mechanical function, diastolic function? Actually, when I looked at the again, I had to prioritize data I had to present today, but full paper hopefully should be coming out soon, where, of course, I recorded function during my CGP experiment. There was no change during this short period of time when I was using CGP. There was no significant change in function. Beautiful talk. I was wondering on the possibility of, thank you. I was wondering on the possibility of depletion of glycogen stores. Yeah, good question. Maybe linked to increase in ketone utilization. Yes, and this is really a basic metabolism planning failure. I haven't measured glycogen stores. Probably for the first time in my life in a set of metabolism experiments, I haven't extracted and measured glycogen in the hearts. So whether some of the glucose that we see in terms of enhanced utilization is coming from locally broken down glycogen, you're right, that could be the case. But we don't have a case of, we don't have ischemia, so we don't have that type of stress that could be driving increase of glycogen stores. But saying that, I haven't measured glycogen stores in these hearts, so I can't answer that. So that is definitely another metabolic assessment that could have been done in the context of trying to decipher impact of sodium on substrate metabolism. You mentioned the lactate is upregulated in the heart failure models. No, actually, what I said is that when I did perfusions in other models of hypertrophy and failure using rats, different models that I have done, and I've done C13 substrate utilization studies, I found that even though in literature we talk about fatty acids versus glucose utilization, I actually found that Lankendorf perfused rodent hearts really like lactate as a source of carbohydrate. And I don't think I'm alone in this observation, because I think there's been studies, published studies from some of the seminal names in literature that has shown a similar carbohydrate preference for lactate of perfused rodent hearts. So just to be clear, I have seen this trend in both my sham control groups and various animal models that I've done C13 substrate utilization experiments for. Does that mean that glycolysis is the part of glucose metabolism rather than mitochondrial glucose oxidation that is upregulated in the failing heart? Kutuba, I'm still struggling to understand the question. In addition, or add anything to what I've already answered in your previous question further up. So I'm sorry, I'm not sure what this question, last question, is asking of me.
I mean, you've gone through 27 questions, which is fantastic, and I think again it just shows how much possibility there is to engage here, providing the people are willing to stay and the speakers are willing to stay on. And it seems like most of the people are still on, listening to this.
Thank you, everyone.
There's also a separate discussion clearly going on between Mike and David Eisner in the background, which is great, because I think David asked a really important question, which is quite tricky to answer. But, maybe it comes down to, if you even get an activator for the sodium pump, you wouldn't necessarily use it to activate 100% of the pumps. Just like when you have digoxin, you don't inhibit 100% of the pumps either. Maybe that drug would need to be titrated. So maybe that's where you start achieving clinical relevance, rather than killing a patient by overactivation of the pump by reducing contractility, or over inhibition of the pump by essentially raising calcium and killing the patient from arrhythmias. So maybe it's a titration thing. I don't know.
Actually, Davor, I have one concern. Given that my recent ex-boss, Mike Shattuck, is battling this question on my behalf that I didn't answer. I'm also now concerned that my PhD supervisor is watching this webinar and that I haven't mentioned glycogen. So Marie, if you're watching, I'm really sorry. That was epic failure on my part on designing my metabolism experiments. I definitely have to go back to the lab and do that.
Well, I think it does raise a lot of questions, which is a sign of good studies and good research.
No, so I'm very grateful. I'm very grateful to everyone. I'm sorry if I couldn't understand a couple of questions. I apologize. If people want to get in touch with me, I'm pretty sure my details are shared. So if anyone wants any further questions to ask, I'm more than happy to engage in discussion.
There was, okay. So I mean, there was one other question from, or maybe a comment from Matthias on the Q&A, but I think that's probably more just a comment.
Yeah, about elevated lactate in acute, yeah.
So I think first of all, Dunja, I'd like to thank you for sticking to the time really well, allowing lots of time for Q&A, which is clearly quite engaging, and actually for being brave enough to actually combat all these questions head on. And so I would just like to say that now so in terms of housekeeping, all the slots are now filled for the seminar series. And we'll continue on Thursday, and we basically then will see you there again. But I would just say that I would re-advertise again the talk for Thursday. The talk on Thursday is by, and hopefully I'll be able to share my screen now so that you can see me. There we go, right. Okay, so the talk on Thursday is by Professor Mike Shattuck. So he may be able to answer some more of these questions in person as well. And of course, I'd like to thank the ISHR again for support. And obviously, any of you who are not members, you get a lot of information if you just go on their website. And again, I would probably also say that on Friday, we have Dr. Susmita Sahu from Icahn School of Medicine. And so we're going to have a busy week, even though it's an Easter holiday, pretty much starting for most people. So once again, thank you, everyone. Dunja, thank you very much for your time and effort. And and see you all on Thursday again, hopefully.
Thank you. Stay safe, everyone. Thank you very much for this opportunity one more time. Thank you.